All right, so GWAS type of studies before Human Genome Project or the GWAS studies, it was kind of done in animals. These are done using mouse strains. So if you look at the lab rats, there are usually two major, like there's bulb C, there's B6, the major two strains, and if you mix them, you get a heterozygous. But for this heterozygous F1, if every generation you do litter mate, just sibling mating again from the same parent, exact same parents, siblings, you mate them again, and you do this over and over again, always mate the litter mate with each other. You do this for the next, say, 20 generations. What you will end up with are mice that are fairly pure. They become more homozygous. But then if you look at all the different mice, out of the 10, 20 different generations of mice, their genotype are quite different, like this. So each mouse is heterozygous, but between different mice, you can see their genome are quite different. Some locus are inherited from the first strain of mouse. The other locus might be inherited from another allele or another strain of the mouse. And so, so this genotype is H or this genotype is B. And so, the F1 offsprings are identical. F2 offsprings are different, but then you just keep crossing. And then at the end, you'll get a lot of these very, very homozygous locus. And so at this time, we can then do genotyping. It's like a mouse genotyping on each location. For example, at one location, like one locus, you can ask how many of these offspring mice have the H allele, how many of them has the B allele. And you can just look at their distribution on a particular gene, and then you ask, so this is the SNP for gene X, right? And these are the B allele, the H allele, B, HH, and so on. And then you try to associate with some phenotype. For example, obesity. And you say, oh, look, every time we have a B allele, this mouse is fat. But every time we have the H allele, the mouse is thin. And that can give you some associations. Do you need to do multiple hypothesis testing? Sam? Yes, well, it's because in this particular case, we only look at gene X, but in reality is you sequence the mouse, you do the SNP typing of the mouse, all the SNPs in this mouse strain. And also we are only looking at obesity, but you can imagine we can look at their fur color, we can look at their eye color, we can look at some other whatever phenotypes, right? So definitely multiple hypothesis testing. So this is called a quantitative trait. Like a fat or a thin could be a binary, but you can also look at some traits that are quantitative. Say plant, you can look at how tall the plant, the rice or the Arabidopsis is, right? Or in the mouse, you can say, instead of say fat versus lean, you can say how heavy it is. Then it will be a continuous value. So that's a quantitative trait. And then you can see whether there are some associations between the genotype and the phenotype. But also we can identify RNA. Supposedly we take their like maybe fat or some muscle or blood, and you measure the expression. Then you can also do these associations, right? Yeah, so you can link the expression level of some gene with the genotype of some other genes. Could be the same gene or could be different genes. And so when we do this, do we have multiple hypothesis testing? Yes, we do, because here for genotyping, we tested many genes, and also for expression, if you do RNA expression of the mouse blood, it's many genes together. And for every gene, you test whether the genotype and the expression level has associations, right? So you definitely have multiple hypothesis testing. And so, so in this situation, it's called the eQTL mapping. Basically, so many disease-associated genes have been mapped with QTL. So these are really linked to a phenotype. But eQTL mapping is, we don't know whether the expression level is related to a disease, but at least we know that we see observe the expression differences on a particular gene. And so we are considering the expression of a particular gene as kind of an intermediate phenotype, right? We say the transcript abundance can act as an intermediate phenotype between the genetic locus and the clinical phenotype. And so we can now incorporate information of genotype, expression, and some clinical trait to construct the regulatory networks and improve understanding of the disease. So for example, we look at this is the SNP level. And you look at, you can see here, this is one SNP, the mouse with one particular SNP and the mice with another SNP. And you look at the expression of one gene, these with genotype zeros all have high expression, but the genotype 1 mice all have lower expression. That is eQTL analysis. Very often you will find some SNPs to be related to the expression of some genes. And that's very, very important in mouse studies. Can we do eQTLs in human? Can you do inbred mice or inbred human? You can't do that, right? So you can only do this in just big populations, and this has been done. So there is a project called the GTEx. They have collected, I have to remember, I think close to 1,000 individuals. And so these are actually cadavers, people who died, sometimes not from natural causes, could be an accident, just like they decided to donate their body. And they took out the different tissues, organs, and then they did, first you can do genotyping. So we know the SNPs or the genome sequence of those individuals, but also depending on the tissues, organ that get donated, you can also test their gene expression across different tissues. And so now we can use, expression again as a phenotype. Then we ask, are there specific SNPs? So because here we are not having, because when you have inbred mice, they are homozygous. At each SNP location, the 2 copies are the same. If you look at here, so you can see here the 2 copies, the mom copy and dad copy, when you do like 20 generation of inbred, they are homozygous. But in human, it's not the case. You will see, so for example, say, like in one population, you might see that, when you have a CC genotype, the expression of some gene might be low. When you have CT, the expression level might be intermediate. And then when you have TT, then this gene expression become very high. And you might kind of observe this in 2 different populations, and that will give you a hint that this particular genotype is linked to the expression of another gene. And so this is a eQTL. Basically, it's a SNP associated with the expression changes of some genes. Okay, and so people have been doing this. They are especially interested in the eQTL, the SNPs that are regulating genes that are close by, say within a megabase of each other, because they think maybe this particular SNP is related to the regulation of the nearby gene. Okay, so this eQTL studies has been ongoing. And you can use it for a number of these different studies or different purposes. For example, you can use it to identify the causal cell type of some complex disease. Like rheumatoid arthritis is some kind of an immune disease. Many different cell types have been implicated over the years. Like people don't know what cell type is relevant. Because if you look at our, you take our blood, within there's the red blood cell, there's neutrophil, dendritic cell, macrophage, CD4, CD8, B cells, and there is also the Th1, Th2, Treg, Th17, depending on how detailed you want to separate. Some people might think we have 100 different immune cell types in our body. And so if it's kind of an immune-related disease, which is the cell type that really is related to the disease, it's sometimes difficult to establish the causal cell types for that complex phenotype in human. And so there you can use the expression data because there are a lot of comprehensive public available data. This was done in mouse, but you can imagine people can also do this now, especially potentially in single cell now. But this is one of the studies where they took many mice, and they also get their initially it was their stem cells, but you can imagine either they sort the immune cells or they can differentiate the stem cells into different neuroprogenitor cells, differentiate into other cell types or blood cell types. Then they can look at the expression level of the different genes in each different cell types. So supposedly there is a study already that has different immune cells sorted from mouse, and they have expression profile than all of them. So the question is, which GWAS SNP, like if you know rheumatoid arthritis, people have already done GWAS, they know which SNPs are associated with RA, the rheumatoid arthritis, but they ask how many, so in this study, there are more than 250 different cell types related to blood. Which one is the one related to rheumatoid arthritis? I can show you how one study solved this question. So they do it like this. So for every gene, you look at this gene expression level normalized by, so they have 250 gene expression profiles for 250 different blood cell types. And so for each gene, you can normalize the expression of this gene by all the other cell types. You want to ask, is this gene cell type specific, right? What is the expression of this gene in this cell type relative to all the other cell types? So some genes might be kind of generally expressed. Other genes might be very specific, say for CD8 T cells, or for Treg cells, or for Th17 cells, and so on. So that's the first one. The second one is for each particular tissue, you ask, let's look at all those GWAS SNPs related to the rheumatoid arthritis. Each of them is a SNP. It's a LD haplotype block. You say, look at all the genes that are in linkage disequilibrium with my GWAS SNP within this block. And we ask, which is the gene that's most specifically highly expressed in one tissue, in let's see, tissue A. And so basically, for example, you have a number of these genes, and you pick the most specific one for one locus. And for another locus associated with RA, you also pick the most specific to that tissue. And then you took all of them, then you ask, what is the level of specific expression for that tissue? And you try this for every tissue. And finally, the tissue that has, all the GWAS SNP that's linked to a gene uniquely expressed in that cell type, that is probably the original cell type of origin for the rheumatoid arthritis. Okay? Yeah, so in order to see, to get the p-value distribution, you want to see whether it's statistically significant, you can just randomly pick some unrelated SNPs. You randomly pick other, say, supposedly rheumatoid arthritis have, say, 50 SNPs. If you pick random 50 SNPs and do the analysis, you can see what do you get, then you will know whether the specificity is good. And so in this particular study, they look at all the different blood cell types, and they ask, which of that cell type give you the cell type specific gene expression. Also, so here it's 39 different rheumatoid arthritis risk loci, and they implicating 170 genes in aggregate. And you can see most of them are related to CD4 T cells. And so you can see, okay, maybe CD4 T cell is related to rheumatoid arthritis. And for a similar approach, you can see that the lupus is actually related to B cells. Okay, so yeah, in summary, like this genome, we have many SNPs, and the SNPs form these LD blocks or haplotype blocks. And so in early SNP array GWAS studies, you only need to tag specific SNPs. And for GWAS studies, you can have either family-based association studies to look at which allele get transmitted to the affected child, or you can do case control studies to see whether there is allele transmission difference between the case and the controls. And in order to really increase the reproducibility, you want to make sure you have big cohorts to do these GWAS studies. Make sure you reduce population stratification and also remove potential family members from contaminating your study. And so now people are also doing these eQTL studies in human. Early on was in kind of mouse, but now you can also do it in big populations to identify SNPs associated with differential gene expression. And they can also use the cell type specific gene expression to identify disease tissue of origin. Okay, so sorry, it's a little over time.